Okay, Bio 1 folks, this is the start of Chapter 18. This is Unit 6, our last unit for the semester. And we spent last chapter talking about how we got from DNA to proteins. And now we're going to spend an entire chapter on how that whole process is regulated, how the cell has evolved processes to regulate it. And this chapter is called Gene Expression. And the cover of your chapter has a picture of one of my favorite fish. The genus species is called Anableps, otherwise known as Quattro ojos, or the four-eyed fish. They occur in Central and South America, and they truly do have four different lenses. They have two eyes that sit above the surface of the water and two that sit below the surface of the water. And one of the reasons they're one of my favorite fish is because I used to have a display of them at the aquarium where I worked, and so I took care of these guys on a regular basis. So these guys can see both above and below the water. SeaWorld, before it built its infamous roller coaster, had a great display of these fish, and they actually had a mechanical fly on a wire that would fly around. And it was a friend of mine that designed the exhibit, and every 20 minutes or so, the fly would go across the water, and if the fish was able to hit the mechanical fly with a spout of water, because that's how they fish, is they look for insects to fly above the water, and then they shoot a stream of water to get the insect to drop into the water, and then they eat it. And so a friend of mine had designed an exhibit so that it had a mechanical fly that would move across the surface of the water, and if the fish were successful in hitting the mechanical fly with a stream of water, it would drop some pellets of food into it. So they actually trained the fish to do that on command, which was pretty cool. So enough storytelling. That's the four-eyed fish. And it's kind of a good example of how genes for the eyes are controlled in this particular fish. So let's first talk about gene expression in prokaryotes. It differs a little bit whether the DNA is in a prokaryote or a eukaryote. And so the first biggest example we're going to talk about regarding the prokaryotes is E. coli and tryptophan in the human gut. Now let's talk a little bit about that. We haven't talked about genus and species, but E. coli is short for Escherichia coli, and this is the bacteria that lives in your gut. It is responsible for helping your gut out. It's one of many species of bacteria in your gut, but it's a fairly common species. And tryptophan is a protein that is in your gut, and turkey is high in tryptophan. Okay, so E. coli normally, that live in your gut, they normally get their nutrients from the surrounding. And if you eat something with tryptophan in it, then the E. coli gets the tryptophan from your diet. But what happens if you don't eat something with tryptophan in it? Then the bacteria can actually produce it. But that requires energy. So if tryptophan is in your gut, the bacteria saves energy by not producing tryptophan and absorbs it from the surroundings. So this is kind of what's called a feedback inhibition. If tryptophan is there, then the bacteria doesn't have to produce it. If the tryptophan isn't there, then the bacteria has to produce it. And this picture is pictured on page 364 of your books. Okay, so take a minute to look at this picture. You can see on the right-hand side those little blue striped pieces. Those are the genes that code for the enzymes of tryptophan. Okay, so there's trip A, starting at the bottom, there's trip A, B, C, D, and E. So that is 5 genes, okay, that encode for these enzymes 1, 2, and 3. And okay, so let's take a look at how the bacteria E. coli regulates the gene expression involved with this tryptophan. It can adjust the level of activity of the enzymes that are already produced. So in other words, a product of the metabolic pathway inhibits one of the enzymes in that pathway. The other possibility is that it can stop the production of the enzyme at the transcription step. Okay, so it can regulate the gene. So how do bacteria go about doing this? So the example that we're going to discuss, like I discussed earlier, is the bacterial cells that make tryptophan, which is a type of protein and an amino acid. And that the way bacterial cells produce tryptophan is through a 3-enzyme process. And we saw that in the previous slide. And remember that those 3 enzymes, the production of those 3 enzymes is caused by 5 different genes. Okay, so these 3 enzymes are made of 5 proteins, and you can see those 5 proteins, A, B, C, D, and E, and you can see the 5 genes in the piece of DNA. In addition to that, you can see an operator, that golden piece, that is a piece of DNA that is the on or off switch for that operon. And we'll get into what the operon is in just a minute. This is pictured on page 365 of your books. And so take a look at that. We have the RNA polymerase. Okay, remember we're going to be undergoing transcription to make it. And we have the operator, and then we have those 5 genes. So why might all these genes used in this one pathway be located close together on the same piece of DNA? That is because all 5 can be turned on and transcribed all at once, or all 5 can also be turned off and none are transcribed. Remember, by transcribed, we're talking about the process of transcription. So let's get into the weeds on this operator. The operator is a section of DNA inside or just after the promoter, and it controls whether or not that RNA polymerase can bind to the DNA. And therefore, if that RNA polymerase can't bind, then you're not going to have transcription happen. So here we get into the definition, and you are going to have to know this, of what an operon is. And an operon consists of 3 pieces of DNA. It consists of the promoter piece of DNA, it consists of the operator piece of DNA, and then all the genes that are involved. Okay, so the operon equals the promoter, the operator, and the genes. So let's talk about repressors. A section of DNA that's further away from this stretch of DNA codes for a repressor protein. And in this case, this repressor protein is going to be a tryptophan repressor protein. And what happens is that repressor blocks the attachment of RNA polymerase. And you can see the repressor protein in red on that picture. So the job of a repressor protein is to bind to the operator and stop RNA polymerase from binding to the DNA. So there's the operator site. Remember, that's that golden piece of DNA that we talked about earlier. And if that red protein, that red repressor binds there, then RNA polymerase is blocked from binding to the DNA. So will transcription take place if RNA polymerase cannot bind to the DNA? No, it won't. So the protein is activated by the presence of tryptophan. So if you take a look, that golden two-piece molecule that's attached to the red repressor, that is the molecule tryptophan. So in other words, then that gene is not going to make tryptophan if that red protein is there. So here is that picture blown up, and this is on page 365 of your books. So the red protein is the repressor, and then the tryptophan that fits into that protein is called the co-repressor. Okay, so if tryptophan fits into that protein, then that protein repressor binds into the operator, the golden piece of DNA up there, then transcription is not going to happen because that gray blob, the RNA polymerase, can't bind to the DNA. And again, that's pictured on page 365 of your books. So that is a repressible operon, and that's a type of negative gene regulation. Repressible operons are usually on the operator site, but they can also be inhibited when specific molecules interact with the regulatory protein, such as we just discussed. And the regulatory protein in this case is tryptophan. Then there's another type of negative gene regulation called inducible operons, and those are usually off at the operator site. And they can be turned on when specific molecules interact with the regulatory protein. So the example that we've talked about for the repressible operon is the tryptophan operon. We're now going to talk about an inducible operon called the lac operon. So inducible operons in general are similar to repressible operons in that they have a promoter, operon, operator, and genes. They also have a protein that attaches to the operator that inhibits RNA polymerase. So in that case, they're very much like repressible operons. So we're going to talk about the lac operon in E. coli. Okay, and these genes code for 3 enzymes that break down lactose. And if you remember, lactose is milk sugar, and lactose can be broken down into glucose and galactose. And this picture is found on page 366 of your books. So again, this is an example of an inducible operon. So let's take a look at how these inducible operons work. In this case, we're looking at the lac operon of E. coli. And in this case, they're a little bit different than repressible operons in that the repressor protein is produced in its active form and always binds to the operator. However, the repressor protein is deactivated when it interacts with a specific molecule. Okay, so in our example, this molecule of the repressor protein or the specific molecule is going to be allolactose. That's going to be the repressor molecule. So let's take a look at how this lac operon works. This is pictured on page 366 of your books. So basically, one easy way to remember the big picture of this is that if lactose, or in this case allolactose, which is an isomer of lactose, if lactose is present, the gene is on. If lactose is not present, then the gene is repressed. So repressor, in this case, inhibits the transcription of the lac operand. If you have lactose plus the repressor doesn't bind, and then the genes are transcribed. Okay, so it takes a little bit to figure out what's going on here, and we can get into double negatives in this case. So just if lactose is present, okay, the gene is on. If lactose is not present, then it's not going to be turned on, because these operands, these genes, are to make the enzymes that digest lactose. So here's an animation that might help you understand it a little bit better. So the uses for both types of negative gene regulation, and remember, we're talking about repressible and inducible operands in this case. So the repressible genes code for enzymes that use anabolic pathways to build a final product. How does using repressible genes for anabolic pathways save the bacteria energy? The molecule is built until enough is present to interfere with the original gene transcription so that no extra protein molecules are made. That's repressible genes. Then we just got to talking about the inducible genes code for enzymes that use catabolic pathways to break down a molecule into smaller parts. And that only produces the proteins if the molecule that is being broken down is present. So in other words, if lactose is present, then the gene is going to produce lactase, the enzyme that breaks lactose down. So let's talk about negative versus positive. Repressible and inducible genes are subject to negative gene regulation. They are always on at the promoter site because RNA polymerase is always able to bind to the promoter site, and the gene regulation turns them off at the operator site by blocking the RNA polymerase from transcribing the gene. So you've got that picture on page 366 of your books. Okay, so in both of these cases, the RNA polymerase is blocked. So positive gene regulation is where the genes are always off, and then the gene regulation turns them on. So again, it's positive regulation, the genes get turned on. So now let's talk about gene expression in eukaryotes. Both of those examples that we just talked about, the tryptophan and the lactose, are both examples that are found in prokaryotes. Let's talk about how gene expression is controlled in eukaryotes. Humans express the gene that makes lactase in varying amounts during their lives. The gene that codes for lactase, okay, is the same gene from our earlier example of negative gene control. In this case, the promoter site has a low affinity for RNA polymerase, so that RNA polymerase has a hard time binding to the promoter site. Because RNA has a hard time binding to the promoter site, this gene is considered off. Okay? In order to turn on the lactase gene, another molecule, CAP, remember our previous example was in prokaryotes, now we're talking about in eukaryotes. So in eukaryotes, they have a molecule called CAP that must bind to the promoter and then attract the RNA polymerase to bind to the promoter as well. In the presence of glucose, CAP is inactive and cannot bind to the promoter. Okay, this is pictured on page 367 of your books. In the absence of glucose, CMAP or CAMP builds up, It attaches to cap and activates it so the cap can bind to the promoter, and then that attracts RNA polymerase. So, kind of summary, and this is found on page 368 of your books, is that eukaryotes have many options for gene regulations. Specific human cells, such as muscle, skin, and bone, actively use less than 20% of their genes at any time. By using different genes, different cells can perform different functions. And again, this is pictured on page 368 of your books. So now let's skip a few pages. We're going to skip to page 379 of your books, and we're going to specifically talk about how eukaryotes go through going from a zygote to a fully functional organism. And you can see that fertilized egg. Remember, that fertilized egg undergoes mitosis, and eventually it's going to undergo mitosis until we get a baby. So as an organism goes from a zygote to a fully functional organism, it goes through 3 processes. The first process is cell division, and that is where that fertilized egg undergoes mitosis. And then the cells produced via mitosis undergo cell differentiation, and those cells become specialized for different functions. And then ultimately, the cells undergo something called morphogenesis, and that's where those different cells become organized into different tissues. So eventually, those cells will become heart cells or muscle cells or liver cells, and those will be organized into tissues. So the materials in the egg direct this 3-step process. And this is discussed, this picture is found on page 380 of your books. Okay, so the proteins, RNA, and organelles from the mother's DNA are distributed unevenly throughout the cytoplasm. So there are clusters of these organelles and proteins and RNA. Any of these substances that influence early embryo development are called cytoplasmic determinants. So these different molecules that are directing this process are called cytoplasmic determinants. And as the cells divide, these cytoplasmic determinants are distributed unevenly to the daughter cells. And so here you can see 2 different molecules of cytoplasmic determinants, kind of the salmon or brownish colored circles and the green triangles. Those are the cytoplasmic determinants. As the embryo grows, cells in one area will affect the gene regulation of cells in another area. And so we know that cells are sending signals to other cells. So cell surface proteins on one cell may be read by an adjacent cell. There may also be growth factors that are secreted by a cell and absorbed by another cell. So these signaling molecules start the receiving cell on a development path that will end with that cell becoming a specific type of cell. So again, with the plasma membrane and with those proteins that are bound in the plasma membrane, there's all sorts of cell signaling that's going on between the cells during this process. So let's talk about early cell types. We have indeterminate cells, and those are early embryonic cells that have the ability to become any cell type. Now, we have specifically called these stem cells, and you may have heard of lots of controversy about stem cells. Lots of folks want to do research with stem cells because these are indeterminate cells. We could manipulate those and grow new hearts and grow new livers for people and grow new lungs for people in the laboratory if we could figure out how to manipulate these stem cells. Then there are determinate cells, and those are older embryonic cells which have already received these molecules that are regulating their genes, and they've already become a specific cell type. So in the natural environment, these cells cannot be, their path cannot be changed. So an example are muscle cells, and these are talked about on page 382 of your books. They start out as embryonic precursor cells, indeterminate cells, and then signals from the nearby cells lead to the activation of a master gene called MyoD, and that cell it then becomes determined to be a muscle cell. The MyoD gene makes the protein MyoD. So here's the example. So the protein MyoD activates both the active gene to produce more MyoD and other genes that produce secondary transcription factors. And this is found or discussed on page 382 of your books. So the secondary transcription factors then activate genes that produce myosin and actin, and those proteins are unique to muscle cells. They also activate genes that produce other muscle proteins and proteins that stop the cells from dividing. Because in order to be specialized, these muscle cells have to stop dividing to form muscle fibers. And so again, that's pictured on page 382 of your books. It's a half-page graphic. So how do we go into a body plan? The signals from the cell cytoplasm and from other cells determine the top, the bottom, the front, left side, right side of the embryo early in development. So there's all sorts of coordination as to what these cells are doing, and there is communication between the cells and chemical signaling between the cells. And so here's an animation of how that works in eukaryotes. So this discussion concludes chapter 18, the gene expression chapter. Up next is going to be chapter 20, the biotechnology chapter. And hopefully you got a good idea with the Labster lab activity that you did with that. We're actually doing 2 Labster activities on that. And just be aware that we are going to cover chapter 19, the viruses, but that's going to be covered in the second half of this course in Bio 2. So we're skipping it for now, but we'll come back to it later. As always, let me know if you have any questions. Have a great day.